Welcome to Read Science, where my co-host Jeff and I interview and just chat with authors of popular science book and engage in other types of science writing and try to understand how they make these topics that are oftentimes complicated, compelling and engaging and understandable to the general public. Today we are doing our Genomics edition, and we are so excited to have here three writers that we can count on to explain the field of genomics to us. First of all, I'd like to introduce Misha Angrist. Misha, would you say hi?
Hi, thanks for having me.
So Misha is the author of the book, Here is a Human Being. This is the hardback. You know what? I got this from the library because I lent my other copies to my students. So, whoops. But that's good. I'm getting the word out. So, Misha, you are an assistant professor of the practice. Interesting. For the Institute of Genome Sciences and Policy. And you're interested in the intersection of genomes and society, especially as it is manifest in the nascent personal genomics movement. And of course, this makes sense because what precipitated the writing of this book was the fact that you are Personal Genome Project subject number 4. And we will get into more details about that and your book there in just a few minutes. I'd also, it's my great pleasure to introduce Matthew Herper, who is a prolific and incredible writer over at Forbes. And I'm looking at his description here on his Forbes page, and it says, I cover science and medicine and believe this is biology's century. He's covered science and medicine for Forbes from the Human Genome Project through Vioxx to the blossoming DNA technology changing the world today. And I'm hopefully won't embarrass you by saying when I tell my genomics students to read your articles, I keep referring to you as the incomparable Matthew Herper. So would you say hi, please?
Hi.
So, yeah, they're going to think, you know, I'm a fangirl, you know, because I say, oh, be sure to read this. Oh, look, a new article by Matthew. So, because I don't think a lot of them are on Twitter and all that stuff, so it's my
He is incomparable.
Yes, definitely. So, and then I'm so excited
That can be good or bad, Misha.
Yeah, exactly.
Oh.
Well, it's in the good way. So I'd also like to introduce Kevin Davies, Kevin A. Davies, and you have to look it up by author because I found there's also a Kevin Davies who's a footballer, right?
That's right.
And I was like, well, that doesn't look like the picture on Twitter. So Kevin is, he was the founding editor of the first spin-off journal of Nature called Nature Genetics. How many years ago was that, Kevin?
21.
21, that's amazing. Okay, and so he then moved on to be the editor of BioIT World. He's written several books, and the book we are looking at today, or actually he is now a publisher of Chemical and Engineering News, and he is the author of this book called The $1,000 Genome, The Revolution in DNA Sequencing and the New Era of Personalized Medicine. He wrote the book Cracking the Genome, and also the book Breakthrough. I couldn't find the name right away, but it's about the discovery of the BRCA1 gene. Kevin, I also discovered, has helped with movie science to help hopefully the movie about the BRCA1 gene called Decoding Annie Parker to explain the science very clearly to the audience. So
I tried.
We will talk about whether they listen to you or not. So
if we can get some tips for improving the dismal nature of science in movies, that would be very handy.
Yeah, we could spend 60 minutes just on that topic, but we'll get there. We'll get there.
Yeah, we'll have to do a show on that, I think.
So, Jeff, why don't we let you, since I've done all the talking up to now, we'll
let
you ask the first question.
I'm trying not to be intimidating. Look at all this, biological power here. Since I am the physical scientist in the room who has never come closer to anything living than helium, I get to ask as we said, I get to ask the Dr. John Watson, and this is Watson as in Sherlock Holmes and not Watson as in DNA, which is easy confusion to make here, although I have always had an interest in X-ray crystallography, which may sound inscrutable, but is related to that. I have two not softball questions, I think, because they are the very big questions here for the person who hasn't done this. And I want to start, since we're calling this our Genomics edition, I said to Joanne a few days ago, could I, without going too far off, call genomics all that quickly becoming vast area of biological science endeavors that involves something to do with sequencing genes? And you can say yes or no. And this is, if we can do this one, the short one, then we'll get to the long question.
Well, they're related, but I think organisms are made up of cells, and cells contain a DNA, and the package of DNA is called the genome. In humans, we have 23 pairs of chromosomes and 3 billion bases of DNA, the familiar 4 letters, A's, C's, T's, and G's. Obviously, for many applications and for many areas of study, we need to sequence the genome. That was what the Genome Project was all about, the Human Genome Project. It was to sequence from the tip of chromosome 1 all the way through to the end of the so-called sex chromosomes, and figure out where the genes were in between and how many they were, and from then on go on and figure out their function. But there are many aspects of applications of genomics. There's epigenomics and quantitative genomics and many other examples that don't necessarily involve sequencing, but the field develops inherently with progress in DNA sequencing. We can talk more about that as we go along.
Okay.
Would either Matthew or Misha like to add to that?
Well, I would just say genomics is kind of industrialized genetics. It's genetics at a much more systematic and some degree industrialized level. And to some degree, it's also a word that looks great on grant applications and funding attempts for companies. So not entirely snarkily. We invent new words so that we can get people to buy things.
Yeah.
Exactly. I would say that maybe at this point, the term genomics, at least in my institution and in my opinion, it feels like a bit of an albatross because we associate it with human germline DNA. Sorry to be speciesist. And of course, we're really interested in organisms, especially humans as a whole, their phenotypes, their epigenomes, their behavior, all of these other things. And so I often feel kind of hamstrung by the term, because I think what genomics at maybe its most useful implies big data. And that's what we're, I would say, probably collectively most interested in.
So there's an interest in having an actual sequence of somebody's DNA, and that's at the root of the next question. But there's an interesting historical convergence here too. It's like, why do one reason that we sequence an entire genome is because we finally can. And so we keep seeing this in the biology discussions that we have, and it's true in the physics discussions and all of science discussions, that when we have new tools, we use those and we look at new things and we see new stuff and exciting things happen. And so finally we get to the dawn of whatever year it was, and we're able to do this. And then that answers part of the question. It's like, okay, it's a very large, exciting project, and I agree to that, to say, let's do an end-to-end sequence of some human genome, and then the question is why? And there are lots of things, the technology and stuff, but what might we someday get out of that? But what I've read interesting and unexpected stuff that people are already learning from doing that. And one thing that we don't learn, for the popular people is, well, once we do that, we don't know where all the genes are. that's a very naive sort of thing to do, but there are many useful and lovely and interesting things. So, first essay question, and starting with Misha over here on my right, would be, what are we getting out of it, and what can we get out of it? What can we look forward to?
Oh boy.
Elementary, my dear Watson, and move on.
I'm glad he started with you, Misha.
Well, I guess the first thing I would say is, I think from the vantage point of 2000 or even 2005, in many ways we have not gotten out of it what we were hoping for. Mm-hmm. And that is the discovery of genes of large effect that mm-hmm. contribute to common diseases like mm-hmm. diabetes, heart disease, schizophrenia, that sort of thing.
Right.
So that has been a disappointment. And if my fellow panelists disagree, I hope they'll get in my face about it. At the same time, what I think we are getting out of it is a much more granular understanding of Mendelian diseases. That is, rare diseases that are caused by mutations of large effect. So there are quite a number of efforts. I'm involved with one at Duke where we are sequencing sick children and in some cases sick adults. Other people are sequencing healthy adults or centenarians, people who have lived very long. And we are finding alleles, that is, versions of genes that are important in these traits. And I think the perhaps the most palpable thing is, in many cases, we're able to end so-called diagnostic odysseys. So this was initiated by the sequencing of this young boy, Nick Volker, in 2009, and finding a causative mutation and actually leading to an effective treatment for his heretofore undiagnosed autoimmune problem.
Recognizing those mutations means you have at least then two sequences to compare with each other. Is that true?
Yes. So there's still a good deal of, I don't want to say controversy, but discussion about how do we know a smoking gun when we see it, that sort of thing. And we're still, I would say, disappointed and too often heartbroken that even when we find something, we don't necessarily have a way to mitigate the child's condition. But I think, judging by the parents I've interacted with, they are universally grateful to at least have something, a diagnosis, the name, and a community of other families going through similar things.
So, Matthew, you can add to that if you want, but Misha brings up the question too of when you feel like you're on the precipice and you finally start a new technique, and this has happened over and over again in various branches of science. Disappointment goes with it because you expect one thing to happen, but then later on you may be vindicated because you'll find out that all sorts of other exciting things have happened instead, and new understandings.
I think there was a difficult the Human Genome Project and the Celera effort were kind of a difficult, back in 2000, were a difficult thing because I think a lot of people knew that that you weren't really going to get that much of medical interest from having a single genome. And all the cases Misha's talking about are cases where we have, this data is really useful when you get a lot of it.
Yeah.
It took DNA sequencing to go from $1 million per person to $1,000 per person before this really starts to come into wide use. I think sometimes we miss the useful stuff that is emerging from these efforts too. I mean, we talk about these rare diseases and diagnostic odysseys. Sometimes a gene from a rare disease can have a huge effect on a common disease. That's how we got statins. They came from the study of a rare disease called FH. And there's also a new set of drugs from a different form of FH, which is a gene caused by a gene called PCSK9. There's also a form of the gene that causes you to have lower cholesterol and a lower risk of heart disease.
Yeah.
And there are drugs for that in late-stage trials. So, I mean, all this biology is slow. We have this kind of sense as we've kind of all been lied to by the PC revolution and by social media and telling them that innovation just happens in some kid in his college dorm room does it. And biology doesn't work like that yet, and may never work like that. But I do think that there's been some noticeable events, and sometimes there's a real lag. And that the Mendelian disease stuff is amazing, but it is kind of also it's ultra rare diseases, and so there are those caveats.
Well, then maybe Kevin, you can add to that, because I think you were the one who mentioned that not only are we talking about technology that I want to get to, but we're talking about big data has an important part to play, and large teams of people, which is not unfamiliar to me with high energy projects and things. But this is understanding these things, and a lot of what swirls around genomics now involves large groups of people, big understanding, big data, and a fair amount of technology right now, right?
Yeah, well, and that's how the Human Genome Project got underway at the beginning. I think for some of your viewers, it may be worth just quickly recapping how we got the first genome before we talk about personal genomes and the thousands upon thousands that are being sequenced every year going forward as the price continues to tumble. And the first Human Genome Project was very much an international consortium, a collaborative effort born by the frustration that people like grad students like myself and Misha at the same time were facing, trying to find a particular important disease gene. It might have been muscular dystrophy or cystic fibrosis that we couldn't find because the maps that we had available were the equivalent of, I don't know, a Google Earth map that just shows the continent of the United States and no detail. You could see a few lakes and a few deserts and a few rivers and a few metropolises, but that was about it. And if you're trying to find a mutation on that scale, you're basically looking for a football field somewhere across the breadth of the United States. So we needed to produce the equivalent of a street map, and so that you could scroll in and zoom in exactly on where the particular problem might be. And that was the effort that Jim Watson, the Nobel laureate, helped persuade Congress, in part because they knew their constituents were increasingly going to be facing these problems. How do I treat my Alzheimer's disease, or how do I save a loved one who's developing cancer? We needed a roadmap. We needed an encyclopedic inventory of the genome. And that got underway, but there was no book in that, really, until Craig Venter, as Matthew just mentioned, launched Celera in 1998. And almost overnight, the whole thing became this fascinating race, because now you had an individual who was sort of mounting a hostile takeover of the Genome Project, saying he could do it years earlier than the planned 15-year timeline, do it much cheaper, basically inviting the academic scientists around the world to go off and do the mouse genome or something like that. Leave it to us, guys. We'll take care of the human genome. But Craig was in a bit of a bind. He wanted to not only run a profitable company by licensing this information to pharmaceutical companies and biotech companies, of which there were more and more being launched, but I think he also ultimately wanted the academic and scientific prestige, perhaps even ultimately a Nobel Prize, that comes with being the person that sequenced the human genome. And you can't have it both ways. You can't keep the data under lock and key to sell it to an exclusive audience, but also make it available. And that's where the Human Genome, they did two things. They stressed that their data, the consortium stressed their data would be free and open to the public. And they also, it took a few weeks to mount the forces, but they got extra funding from the Wellcome Trust and the NIH to basically compete more or less on level terms with what Celera was doing. And interestingly, the company that was bankrolling Celera agreed to sell their sequencing machines to anybody. It would have been a very interesting scenario if they said, you know what, we're just going to keep our machines to our own spin-off company or sister company, Celera, and not share them with the Genome Project. History might have been a little different. And then, of course, I'm sure your viewers are familiar that President Clinton sort of hastily declared a truce and held up the hands of Collins and Venter at the White House in June 2000. And but you know, Craig at that point realized, okay, Celera's not going to make money licensing information because the Genome Project has pretty much trumped us on that front. Maybe we can become a drug discovery company or a proteomics company. And he realized his heart wasn't in that, so he went back to academic pursuits with his institute, and now is pursuing another genomics field, synthetic genomics, launching a company. So that's a brief history of the Genome Project. And there are many books. I did one book, but many, for future people interested in this and future historians, there are many people who've taken, there's no definitive book on this. I'd love to get involved with doing an oral history of the subject. I think that would be fascinating while the folks are still alive. But there are many people who've written, you've got the sort of the liberal Free the Data account from the Sanger Institute in Britain. You've got the Fly on the Wall at Celera account from James Shreve. And you've got many people in between. My book was somewhat sympathetic to both sides, and for better or worse.
I understand that Matthew might be looking for a book project. You could talk to him about that. Does that, from what you say, I'm sorry, but from what you say, Kevin, does it seem fair to say, and I don't mean this in any sort of diminishing way, that we say we will be, before whatever time we will sequence from end to end a human genome, and there it is, and we will make this available. Well, that's a grand thing, and it's a fairly simple thing to say, but is it in a way not such a useful outcome as when Watson promoted all of this, as the fact that in order to do that, there are all sorts of exceedingly useful things we have to know and learn how to do, and this is a good target to promote that.
Absolutely, definitely the latter. It wasn't as if the Human Genome Project overnight transformed drug discovery. I think, as Matthew said, and alluded to.
I want to make a quick note here. The hottest cancer drug on Wall Street right now came out of Celera.
Hey.
I mean, it wasn't actually. I don't think it was actually genomics derived, and it was after Craig, but it was actually a Celera compound that is now at a company called Pharmacyclics. So
that's almost surprising, isn't it?
Yeah. So
something practical.
Is it too late to get in?
It's really too late to get in.
Story of my life.
Okay.
But Jeff, to your question, the Genome Project, it immediately sort of inspired something called the HAT map, where we mapped the extraordinary amount of genetic variation between different populations. Now some 50 million or more, I don't know what the latest number is, specific units of DNA that vary in different people across the world. And of course, it helped inspire the revolution in sequencing technology, because the price tag for the Human Genome Project, that first genome, was about roughly $3 billion spread over 12, 13 years. And now, as we've discussed, you can now sequence a genome for, in terms of the actual chemicals at any rate, just a few $1,000s. Not quite the $1,000 genome, but we're almost there.
Yeah.
Joanne, I have one more very simple question that's related to these. And I want to see if anyone is bold enough to tell me in 2 minutes. I remember that I'm a physicist, so I need the physicist's understanding of how you actually sequence a genome.
Yeah, well, and I think it's important to say what happened between, Venter, the way the NIH, the consortium was originally doing it, and then Venter said, come on, you could do it this way.
And so I'm not looking for the answer that says, well, we use this machine called a sequencer, and we prepare the sample this way. What is going on in there at some level? Because you've got these enormous numbers of things, and the time is certainly part of that. But how do you determine with some precision which of these bases?
Well, I'll very quickly. The technology that was used for the Genome Project was something called Sanger sequencing, and that still has a niche role in some areas, forensics, for example, and is still considered widely the gold standard for many clinical applications. But it has been largely subsumed by next generation sequencing. And there are many different flavors of next gen sequencing. The most popular, most of the labs and companies in the market right now using next gen sequencing rely on one of two systems, one by Illumina, which came out of a British company called Solexa several years ago, and another by Life Technologies, which bought a company pioneered by one Jonathan Rothberg called Ion Torrent Systems. The latter uses essentially is measuring sequencing by pH, which sounds extraordinarily. When I first heard that you could actually sequence DNA by measuring pH, that was incredible. But each time a nucleotide is added to a growing chain of DNA, a proton, or in other words, a hydrogen ion, is released. And if you use some pretty sophisticated technology, and you can detect the release of that ion, you know when a base has been added, and if you know which base was in the mix, you can then compute the sequence. Illumina is essentially taking, looking at the growing sequence of millions upon millions of tiny beads of DNA, each sequence identical in each bead. And as you flow in the chemicals, you use fluorescent dyes to identify which of the four bases has been added. Take lots of photos, you add the bases one at a time, and you build up a sort of a stop motion picture of the sequence. So those technologies have been around now for 3 and maybe 5 or 6 years respectively, and we wait now for the next generation of sequencing technologies, perhaps nanopore sequencing, which will truly break that $1,000 genome barrier.
Yeah, so the nanopore one, I think, when I think of that, it's like as if you asked a little kid, how would you read a piece of DNA? Oh yeah, well, I would just pull it through and go read, and that's what nanopore technology is, right? It's just not as easy as doing that.
I mean, I think the physicist answer might be all these methods use the pairing, the ATGC pairing of DNA to figure out what you're pairing with.
I'm getting that, but it's and we know that's the way we would do it in the the physics textbook, and then we would know that's not the way it actually works. Except that's the way it actually works. It sounds like, and if you can
that's the way it actually works. Sometimes there are colored tags on the DNA, or sometimes there are a lot of we didn't even touch PacBio because it worked completely differently and no one used it.
Yes,
but
right.
It's really cool.
Yeah, but molecular pH measuring is really kind of phenomenal sounding.
Well,
except they have trouble when they're the same DNA letter in a row.
Yes.
So
that is the problem.
Before we leave this subject, at the American Society of Human Genetics meeting last year, I bumped into Kevin, and he said, do you want to come over to the Oxford Nanopore booth with me and see what's going on? And I said, how could I refuse? And so we went over there, and sure enough, it was a real dog and pony show. And they had this little dummy kind of cartridge set up, so the idea being that this thing is about the size of, I don't know, your sunglasses case, maybe smaller, and it's got a USB on it, and you just shove it into your computer. So there was a mob there, but we had this kind of surreal conversation with the CEO. We were asking, how much it cost and how fast the data would be generated and when the data would be available and how accurate it would be. And they weren't really answering any of those questions. So I'm wondering, Kevin and maybe Matthew can update us and let us know where we are.
Yeah,
Are answers to any of those questions available yet?
Well, that's Matthew's job, so I'm going to let him take first crack at that.
No, everyone's still waiting. Everyone's really wondering, but no.
Yeah, I mean, Jeff and Joanne, I think this was Oxford Nanopore really kind of burst onto the public stage about 18 months ago when they presented at a scientific conference in Florida, and the crowd really just went bananas because they were, as you said, Joanne, you're talking about a protein, if I can do the camera thing here. It's a ring, a naturally occurring protein. They've engineered it, and the idea is you simply thread the DNA through under an electrical current, and because the 4 bases differ in size and shape, as they wriggle through that pore, they will block the current by a different amount. And if you can, again, apply some very fancy hidden Markov models and fancy informatics and equate the blockage in current to the pattern of bases going, the sequence of bases going through the pore, bingo, you've got a phenomenally cheap DNA sequencer because you're not using lasers or fancy dyes. And you can, the problem isn't to speed up the sequencing, it's to slow it down so that you can actually make sense of each base one at a time. They had said 18 months ago that they would have launched, the plan was to launch sometime this year in 2013. We've heard nothing about those plans since then. So it does appear they're a little bit behind schedule, but I guess, you know, when they come out with this, particularly with this fancy USB thumb drive sequencer that Misha described, you know, they want to make sure it's right. So, you know, the rest of the field is still, I think, certainly optimistic. Most people are still fairly optimistic that this is going to be worth waiting for, but the wait is getting a little painful right now.
Well, there's a problem every new sequencing company has now, too, which is that Illumina keeps getting better, and everybody's using Illumina, and Illumina is becoming the clinical standard. So you have this problem of becoming a niche. I mean, the thought of Oxford Nanopores, maybe they'll have really long reads, which means instead of most of these technologies chop the DNA into little pieces and you got to puzzle them back together.
Right.
Maybe they'll have really long reads, and that'll be the. But will they initially be able to come out of the box and be better than the thing everybody already has installed? That's hard. I mean, that basically the thing that Rothberg did that was very smart with Ion Torrent with the pH was he made it cheap and he made it something that people who were priced out of DNA sequencing could buy, and instantly sold a lot of machines. It became the main real competitor to Illumina just because he found a niche. So it's kind of almost as much a business problem. Which, by the way, Kevin, have you heard anything about Jonathan now that he's left Life?
No. Tell us, what's he doing?
I have no idea. That's why I'm asking.
and so Jonathan's one of those people, so when you read the history of the Human Genome Project and all these things, there are a lot of scientists who are motivated by very altruistic, charitable, whatever reasons. Like they even take a complete turn in what they were studying, you know, because maybe a family member had something go wrong. I think there's a lot of interest in the personal aspect of these stories of the people involved with the Genome Project and the development of sequencing technology.
I don't think Rothberg's motivations are entirely altruistic, Joanne.
Well,
I think he's
I think right now he's
money as anybody. But,
yeah, he's doing well.
But he did tell the story. He was the founder of Curagen, one of these genomics companies that briefly cashed in on the sort of the fruits of the Genome Project in the 90s, you know, and then entered hard times. But he span off a company called 454 Life Sciences because one of his during the birth of one of his kids, there were some complications, and he claims, I don't know how true or apocryphal this is, but he claims that he was in the NICU waiting room that night, pacing up and down. He needed something light to read, and he clutched into his briefcase and pulled out an IT magazine, and there was a picture of a transistor on the cover, and he said, bingo, that's it, that's what we need for sequencing. We've got to make it smaller, we've got to do it in parallel, and it's a great story, and I trust that it's true.
So let's talk about this just a little bit, this concept of stories and science and things like how the Human Genome Project, when Craig Venter came in, well, he was working with the main project, but he said, no, I'm going to do this differently. Like how this sort of clash actually made the Human Genome Project more interesting. It got journalists excited, it got the general public excited. And what does this do for science overall? And do we always need that to make science interesting to the general public?
Well, I think back, I remember one of the items about the double helix, the race for the double helix that excited people was that there was a race. There was always the threat that Watson and Crick could be scooped by Linus Pauling, or maybe Rosalind Franklin would actually build a model and trump them. Mm-hmm. And like I say, the Genome Project was this boring, pedestrian, humdrum, government-funded project until Venter said, no, I'm going to make it mine. And then, this is obviously pre-blogs and Twitter and so on, all the muckraking happened really in the pages of the New York Times and the Washington Post. Mm-hmm. And it was every week or month, it seemed, there was rival press releases going out. And there was a lot of animosity, there still is, to Venter because of some of the things that he did or was associated with during the 1990s. He was associated during his time at NIH with one of the first efforts to mass patent human genes. Mm-hmm. I don't know that was his choice, but that was the NIH doing that on his behalf. And then, of course, he was one of the first people to cozy up to Wall Street and to VC funding. And through an association with Human Genome Sciences, I remember when the Wall Street Journal, the New York Times published the fact that his stock options were worth about $12 million in, I don't know, this was 1993 or something. And I think people collectively thought, what? You can get that rich just cloning and sequencing genes at random? God, I hate you. And he's, so...
And he has a really
go ahead.
He has a really nice boat, but
he does.
And has always had a really nice boat. But I think something harmful happens from that kind of race conversation, which happened with embryonic stem cells too, which is that everybody who on each side starts promoting the technology as being the be-all and end-all of everything. Mm-hmm. Even when it's not, and that's one of the reasons we had all these stories years later of, oh, the Genome Project didn't do anything. It's because both sides are, that kind of dynamic has the harmful effect of you're not really talking about the technology, and it kind of gets inflated as a side thing. In the same way that we had embryonic stem cells were going to make Christopher Reeve walk again in 10 years. Mm-hmm. Kind of absurd propositions.
I actually appreciate that extra perspective here. Before we go on to a few other things, I do want to ask Misha and anybody else who wants to join in, but I do want to ask about the Personal Genome Project, and then to, why this project? This is George Church. Why this project? Why did you join? And this dawn of being able to get your genome sequenced. And what does that mean for us? Does it mean anything? And if everybody could have an ion torrent and could genome sequence everything, should we? And why don't we, even if we did? And I know the answer, but let you guys explain. So let's start with Misha, because you were actually subject of the project.
Well, if someone gave me an ion torrent for Christmas, I think I'd probably return it or re-gift it.
Re-gift.
Yeah, so Kevin mentioned the 1990s. Actually, Kevin, when he was editor of Nature Genetics, published the linkage paper that was sort of the main bit of my thesis. I was studying a birth defect called Hirschsprung's disease. And I was always struck when I was doing that work as a graduate student and later as a postdoc that genetics was something that even though we all have DNA, there was some firewall between those people who got to do genetics and those people who had genetics done to them. And so, if you didn't have the letters after your name or the white coat, then you had no business looking at your own DNA, let alone anyone else's. Mm-hmm. And so that always kind of rankled me a little bit. What were we sort of hiding? And I remember distinctly in January 2006 when there was this cover story in Scientific American called Genomes for All, written by this guy named George Church at Harvard, saying, you know, the cost is falling and pretty soon we're going to be able to sequence large numbers of people for not that much money, and we ought to talk about what that's going to look like and who's going to have access to that information, which is all well and good. And then he kind of took it a step further, saying, what if we actually had a cohort of people who were willing to just make their own genomes public, no strings. And I thought, boy, this guy is crazy. I have to meet him. And that was sort of the genesis of what became my book. And so, you know, I would say for the first 3 years or so, he was not really taken very seriously. It might be even longer than that, and to this day there are people who don't take him seriously. But he got a cohort of 10 people, and I badgered him until he relented and let me become one of them, who would be willing to do this and see what happened. And then after very patient petitioning of the Harvard IRB and several other hospital IRBs in Boston, he got approval ultimately for 100,000 participants to enroll, and thus far has enrolled, I don't know, 3,000, maybe more by now. And so the basic idea is that these people will have their genome sequenced at no cost, and they will make the data public, and anyone and everyone can have access to those data. But in order to enroll as a participant, you have to pass an exam that covers not only genetics and genomics concepts, but some of the kind of ethical and social questions about what it means to have your genome public, and whether this is something that you're really comfortable with.
I'd like to ask Misha a question.
Yeah, go ahead.
So in your book, you write it's a really moving account of what it's like to get this giant spreadsheet of data and not necessarily know what it means. And I'm wondering if your perspective on that has changed. I mean, I assume you've been continuing to get information from your PGP data since the book came out. What have you learned? Have you learned anything? Either biological or metaphysical, or?
Well, I mean, you know, this is the thing, is, as I said, I'm part of this whole exome sequencing effort at Duke, the exome being the 2% of the genome that codes for proteins. And I think what those families learn from us is much more palpable and moving and meaningful than anything I've learned about my own genome. I think for a generally healthy adult, yeah, there's a possibility that you will learn about your risk for a devastating late onset disorder. But if that does not emerge, then what you're confronted with are some things that are interesting if you've trained as a geneticist the way I have and Kevin has, but probably you are unlikely to learn stuff that is going to radically transform your life or your healthcare. I mean, it's very hard to generalize because even now our sample size is still fairly small, and we do have cases even in the PGP. John Lauerman, who's a reporter for
Bloomberg,
was a PGP participant and found out about a medical condition and sort of went on his own little diagnostic odyssey. And that's certainly bound to happen to some tail of the distribution. But I think for me, I think for most PGP participants, if I'm not being too presumptuous, we want to contribute to science, and we also want to have access to the data that emanates from us. So that's kind of the exchange, I guess. And to the extent that we can perpetuate that and find a cohort and build a cohort of people for whom that is an equitable deal, that's what we're trying to do. I'm not sure if I answered your question.
Yeah, I mean, the science still has a long way to go, but if the technology continues to get cheaper and cheaper, as the founder of 23andMe, Anne Wojcicki, proclaims, there will come a time in the not-too-distant future where everybody is sequenced potentially at birth. We already screen every newborn. We prick their heel. We do this thing called the Guthrie heel stick test and screen for 30, 40, 50 different potentially devastating genetic disorders. Why stop there? Why not, if sequencing becomes less, just a few $100s, why wouldn't we just get the whole sequence and then we can have a sort of the ethical debate, well, do we just hand that CD or that flash drive to the parents when they leave the hospital, or do we put it in some sort of lockbox? That's an interesting discussion. I think for people who aren't about to volunteer for the Personal Genome Project run by George Church, although I would urge them to take a look at it, there are really two ways now that you can get access to your genome. 23andMe, of course, is the one true standing direct-to-consumer genetics company. There were a whole bunch back in the 2007, 2008, but most of them have either been gone, changed business models, or been swallowed up, and the technology's been doing other things. 250,000 or more people have signed up for 23andMe over the past 5 years. They're pushing on for 1 million by the end of this year. That's the goal. And the price has dropped to just $99 and no monthly subscription fees anymore. And you learn a great amount, and it's great fun. I would stress you learn about something about your ancestry, where some of your forefathers may have come from. You learn about your predisposition to different traits, some of which are fairly trivial and fun, some of which are a bit more important, such as the ability to process drugs like statins that Matthew mentioned. And then you learn about whether you carry risk genes for a growing number of diseases, including Alzheimer's disease, including some mutations in the BRCA1 gene, and a whole slew of other genes. So, it's not for everybody, but there is some positive information that you can extract from that. And if you want to go the full in and have your genome sequenced, the company we talked about earlier, Illumina, now runs, I think it's twice a year, a conference where for $5,000, you not only register for the conference, they will sequence your genome in its entirety to gold standard quality, and they'll throw in an iPad with the results and a nice browser as well. So the $5,000 genome is literally here, it's within reach. That's where we are today.
So let me ask one, Matthew, have you had your genome sequenced? Because I know these two guys have. No. So the other thing is, I read both of your books and sort of giggled at the fact that you both bring up the fact that you were balding. I was just like, what?
You didn't need your genome for that, guys.
I know. And so that I thought represented this. Okay, nice. So you're but you know that already. So a lot of us, we just happen to know already, that we've got high blood pressure, or that, Alzheimer's could be a possibility in our future and things like that. So, but I think it would still be interesting. I would be interested in being a PGP participant just because, for science, just for science, know. And I'm thinking, the things I've been through in life, I'm not worried about an insurer not insuring me because they found out I may get something. So let's talk quickly, because I know Matthew needs to leave pretty soon to catch a phone call. But I want to talk about Angelina Jolie. So let's just talk about BRCA1 for probably for the rest of the time. We'll probably take her. So we got Angelina Jolie, we've got the Myriad case, and we do have that movie Decoding Annie that I do want to talk about. So let's talk about Angelina Jolie really quickly, and then segue into Myriad, the Supreme Court case. So Matthew, do you want to start?
Oh, well, I thought Kevin would be the one to give a good description of BRCA.
He would, because he has a book on it.
I think the Angelina Jolie thing, it was a courageous thing to do. I think one of the more important things to watch, it brought a lot of awareness at the same time as the Supreme Court decision on some, but not all, of the patents that Myriad has on BRCA. And also, I think this movement coming out of Randy Scott, who was an old competitor of Craig Venter's back in the genome days, is now running a company called Invitae to free the data to make public equivalents of the private database that Myriad has been able to keep around that gene. Could be something that's really important, if not simply on its own as a thing to watch as we move forward in dealing with this information. I mean, to me, a lot of the issues around BRCA are actually they're settled. It's just that BRCA got grandfathered into the bad way of doing things. I mean, I don't think that any company could build the patent estate around a gene like Myriad did, but that's because the world changes.
Maybe Kevin can give us the quick thumbnail of why it is that we're talking about Angelina Jolie and what the Angelina Jolie thing is and what relationship it has with breasts and movies.
Yeah, no, BRCA1 has been dominating the news. So yeah, Jolie came out and talked bravely about and very candidly about her personal decision to prophylactically have a double mastectomy based on family history and having had the gene test. But the reason the BRCA1 patent went all the way to the Supreme Court is that for the last 15 years or more, Myriad, which ultimately was the first to find the gene in 1994, patented it. That's not unusual. Many academics patent genes. It's what you do with the patent that's the question. And Myriad refused to share or grant access to anybody else to offer testing, full-blown sequencing testing on the genes. So you basically had to go to Myriad if you wanted to sequence BRCA1 and 2. And women, along with other parties, sued on the basis they were denied a second opinion, which I think and most people would say they certainly would have the right to. So the Supreme Court decision to unanimously overturn the actual gene patents was, I think, almost overwhelmingly supported by the scientific and medical community. That afternoon, after the decision came down, at least a couple of genetic diagnostics companies came out with press releases saying, and on Twitter saying, we're going to launch our own test for a fraction of the $3,000 or $4,000 that Myriad charges. But since then, Myriad has announced they are going to sue at least some of those companies because they still have a whole bunch of patents that were not touched by the Supreme Court decision. And a very interesting patent battle is still about to be waged on whether they can still have access to those royalties.
So this means when 23andMe or even the Illumina sequencing happens, can they not even, before the ruling, can they not even say you have the BRCA1?
Well, 23andMe is only looking at, I think, 3 specific variants last time.
Okay.
So they're not doing sequencing.
Not a full sequence of that.
Not a full sequence. And there's a school of thought, I don't know if this would hold up in court, but there's a school of thought, but if you do whole genome sequencing, you take somebody's entire genome, chop it up, and sequence little bits and then put the jigsaw together, since you're not isolating the BRCA1 gene and screening for it in isolation, you're not violating Myriad's pattern. That's certainly that school of thought. But some people will maybe sequence the gene, but then not actually do the analysis for fear of Myriad coming down on them, at least before this decision.
And Misha, you were actually at the Supreme Court that day, were you not?
I was. So people always ask me what that experience was like, and if you remember the famous David Foster Wallace essay where he goes on a cruise ship, it was called "A Supposedly Fun Thing I'll Never Do Again." I got there about 4:30 a.m. and I still almost didn't get in, except for one of my colleagues actually had a press pass and gave me his number so that I could get in. They only let 50 civilians in, so you know, it's not a it's somehow a peculiarly American process to go and watch the Supreme Court argue, but it's not a great shining example of our system of justice, I don't think. And then, you know, of course, you get in there and the justices are using all of these metaphors to describe science, you know, chocolate chip cookies and medicinal plants from the Amazon and all of this stuff. And I was kind of shocked and frightened by all of it. And I give them...
It's well worth digging out the transcript of the oral arguments,
yes,
because they go all over the shop. It really is. Guys, this is the best we can do.
Yeah, exactly.
Terrifying. I gotta go now that
I know. I was going to say thank you. Do you have anything you'd like to add before you disappear?
No, thanks for scaring me, Misha. I appreciate it.
Anytime, buddy.
Thank you, Matthew. I appreciate
Thanks, Matthew.
it. Thank you.
So there's another idea, Joanne. We have to do an episode on science in the courts.
Yeah, oh,
scary.
Well, I know some good people who write about politics and science that could be. Yeah. But I would like to talk about really quickly, because we've got 3 minutes. So I went and watched this movie, Decoding Annie, because I wanted to cover for
Decoding Annie Parker.
Annie Parker, right. I keep shortening it, maybe for Twitter. So like I said, it's the story of this woman who's had cancer 3 times. 2 of them seem to be BRCA1 related. A breast and ovarian. Family members, of course, had died, and so the thinking when this was happening to her, of course, no one wanted to believe that a multifactorial disease like breast cancer could possibly have a genetic component. And there's Mary Claire King, who could see that this is a possibility. So we have this very brave woman, this really engaging story of this woman, but then also woven in with some of the science, including Mary Claire King. And when I watched it, I sort of went, you know, I know these concepts, and I didn't think some of them were explained very well. But I understand from you that you actually were called on to try and help them explain and get the timeline right. And I think even if timelines were wrong, that would go over the heads. But what I felt was more of an egregious error was the fact that this stuff could have actually been explained properly and clearly so people would understand the science a little bit better, but they didn't take... This
doesn't bode well, Joanne, this doesn't bode well since I'm credited as the genetics consultant on the movie.
But it's not, but the thing is, I looked at your notes versus what I remember watching, and I was like, they didn't listen as well. Directors have to, producers, directors have to make decisions.
I think, just for background, this film was shot 18 months ago, nearly 2 years ago now, on a minuscule budget by a highly respected but not terribly well-known director named Steve Bernstein. And he got a tremendous cast. Mary Claire King is played by Helen Hunt.
Yes.
And there are alums from The Office and The West Wing and Breaking Bad. Aaron Paul has a role in it as well. Samantha Morton plays the title role of Annie Parker, who was one of the first women in North America to be tested for the BRCA1 gene. And so I got a call, I don't know, a month or maybe 2 months before they started shooting, from the director who'd found my first book, Breakthrough, gathering dust in some library somewhere, and saying, wow, you wrote about the whole story of how Mary Claire King, throughout the course of the late 70s and all through the 1980s, believed that there might be a gene that explains the predisposition to breast and ovarian cancer in some families, at any rate, when the whole scientific community was highly skeptical. But she stuck to her guns, and sure enough, in 1990, she mapped the genes. So the director of this film has woven in the story of Mary Claire's saga in between the main story, which is the family saga of Annie Parker. But I wrote them and called in a whole series of notes about, you can't do that. You don't have food parties in the middle of the lab. We try to keep food out of the lab, and correcting details. But at every other scene in the lab, they wanted to have these deep philosophical conversations about what oncogenes and tumor suppressor genes were, mixing metaphors. And I tried to say, you know what, they were looking for, this is a race to find a little bit of real estate on the human genome. It really didn't matter what the function of the damn gene was, because we didn't know what the function of the gene was. We didn't know if it was going to be a nuclear protein or a membrane protein or a cell signaling protein. That would all come later.
Right,
so you don't even need
time.
that. You don't even need that. It's simply about finding strategies to get close to that piece of real estate that we now know is on chromosome 17. And they took on board some of my suggestions. Others, they, I guess, because of time or whatever, they decided to ignore. I guess that's how Hollywood works.
I think so too, and I think in the interest of making it a human story, maybe it's okay if you don't get the science right, but
right.
It's sort of, it hurts the heart of science.
I'm shocked to hear you say that, Joanne.
Well, once the film has secured its distribution deal, and maybe you'll touch on this in your review, Joanne. I'd love to sort of come clean and say, this is Confessions of a Science Consultant. That blog is waiting to be written.
Right, and there is actually. So you were lucky to be found where you were found. There is actually the Science and Entertainment Exchange. Is at AAAS, and but there is actual groups out there that want to pair scientists with people making movies. Say, we've got scientists for you. Come use us. Come ask us questions, and we'll help you get the science right.
Yeah.
So I think that's.
Well, I want to give a very quick plug to another movie that I think would really interest this audience if they've stuck with us for the last 60 minutes, which is The Perfect 46, which is I've seen a preview of that. Not released yet, still in the editing room, but a filmmaker named Brett Bonowitz has written a very thoughtful piece about a company not unlike a 23andMe that in the not-too-distant future is able to essentially weed out genetic disease, and raises lots of discussions about eugenics and the perfect race and so on, until, of course, something goes terribly wrong and start to pop up. He doesn't try to explain, and maybe this is a wise decision, not how this could actually happen. So it's more about the sort of philosophical and medical and ethical debate. But I hope he gets funding for that and finds success on the festival circuits over the next 6 to 12 months.
Mm-hmm.
Oh, yeah. So I could show my classes something other than Gattaca.
Right. Yes. Exactly. I did tell Brett there weren't enough car chases for me. There was no action. It needed a little more sex and violence.
Yeah.
But
we want that. So we are over an hour. Not that matters, since we're not like in a time slot that sponsors are paying for anything. But I don't want to hold you guys up. But this is the time where we like to ask. Do you have anything else you would like to add to our viewers, or for archiving forever and ever on YouTube, or however long forever means on YouTube? So, Misha, is there anything you'd like to add that we didn't ask, or maybe you'd like to say?
Well, we touched on it probably an hour ago, just this idea of using narrative to explain science, and obviously with the movie discussion it comes up again. I have a colleague at Duke in philosophy who every time I see him reminds me he's a philosopher of biology, and always takes pains to remind me that stories are not science, Misha. And I you know, I don't disagree with that on its face, but I think stories are absolutely essential to the communication of science, and we need more of that. And in many ways, while we may be in the final throes of science journalism as it was constructed 10 or 20 or 50 years ago, we're really in a golden age, I think, of science communication. And there are so many amazing and instructive and enlightening and entertaining stories about science that remind us about not only the amazing work going on in the lab, but the way this stuff makes its way into the world. Not always the way we would like it, but I can't emphasize enough how important I think it is to tell those stories.
Well said, and I would just like to add, go buy Here is a Human Being by Misha Angrist, and while you're at it, buy the $1,000
and the $1,000 genome.
Here we go, here we go. These are the hardback covers, but they're both out in paperback now, right? So your books came out almost exactly at the same time, didn't they?
Yeah.
Yes.
I don't think I got a paperback edition. Misha has one up on me there.
Oh, okay. Well, I thought I saw, but maybe that was the audio edition or something.
Could be. I did get an audio edition, yes.
Yeah.
I think Joanne and I are readily going to agree with you, Misha, that storytelling is really a core part of what science is about. It's a particular way that we tell stories about understanding the universe, and that's important. I think it's silly to say it's not about stories, because it's maybe not solely about stories, and we have to tell them in the right way, but storytelling is certainly a very important part of it.
Okay, so what I'll do is we'll say goodbye to our audience for now, and you guys sit tight for a second, and thank you guys, everybody out there, for joining us for our genomics edition of Read Science, and we'll be back later with some other fantastic authors to join us.
Thanks, Joanne.
Thanks, Joanne.
Thanks, Jeff.
Thank you, guys.
Jeff. Bye.
Bye.